this specific immune response. So on to the next slide, we're going to take a look at how exactly our body is going to make antibodies. Specifically, what exactly are these different cells doing and how are we going to make antibodies to help protect us? So as we go through this slide, I want you to be filling out your notes. I'm not sure, it's right in your notes packet. So you're going to be filling out the notes that correlate with this. So first and foremost, what is our body going to do when that pathogen now reaches the third line of defense? Our first thing that we're going to be taking a look at is how an antigen presentation and T lymphocyte activation. So let's go step by step. First, a phagocyte is going to eat the bacteria or the pathogen, right? The invader. The phagocyte is going to eat it. So the phagocyte is still playing a role in our specific immune response. Number two, parts of the bacteria, the antigen, goes to the surface of the phagocyte now. So now that the phagocyte has engulfed the pathogen, the phagocyte is now going to have these antigens on the outside of it. So a nice cute little helper T cell is going to come along, and the helper T cell is now going to be activated. And remember, T cells are going to help with recognizing antigens. So let's go to this overview right here. Helper T cells are the major driving force and the main regulators of the immune defense. Their primary task is to now activate B cells and killer T cells. So let's now look at B. So we went through all of those. We're now going to take a look at what these T cells are going to activate. So now we're going to take a look at B lymphocyte activation. The first thing is that the B cell is going to find an antigen which matches its receptors. Now remember, the B cells are producing specific antibodies. So the specific B cell that matches with those antigens on the surface are going to bind to that bacteria. So we have a B cell is now going to use its receptors to bind to the bacteria. Number two, it's going to wait until it's activated by a helper T cell. So it's going to hang on for a second until that helper T cell tells it to be activated. So let's read our overview over here. The B cell searches for antigen matching its receptors. If it finds such antigen, it connects to it, but the B cell needs proteins produced by helper T cells to become fully activated. So it's all connected. Let's head on over to the next slide. Now let's take a look at the plasma cells and antibody production in particular. So what's the next step that our body is going to take? So we now know that our B cell has to be activated by our T cell. So what is that B cell now going to do? Let's look at number three. The B cell is going to divide to produce plasma and memory cells. So you can see here, we have plasma cells being produced and we have memory cells being produced out of that B cell that has been activated for that specific antigen. Our next step, we're going to take a look at the plasma cells again. Whoops. The plasma cells are going to produce antibodies that attach to the current type of invader. So our plasma cells are going to make a bunch of antibodies and shoot them out to destroy the bacteria or the pathogen or the invader that the body has noticed. So let's take a look at our overview over here. Plasma cells are specialized in producing a specific antibody that matched the B cell receptor. So plasma cells can produce tens of thousands of antibodies per second. Kind of crazy. So they're the ones who are making the antibodies to protect the body against this bacteria. Now let's take a look, pathogen destruction and the formation of memory cells. Okay, so we've now made antibodies by that plasma cell. What's the next step? What is our body going to do next? They've released the antibodies that have, you can see in the picture up here, the antibodies are connected to the bacteria. So now that happens, how are we going to get rid of the bacteria? So now we have eater cells. And eater cells prefer intruders marked with antibodies, and they're going to eat them or engulf them. So in the picture here, you can see that we have this eater cell, okay? And now that this bacteria has been binded with the antibody based on the antigen, these eater cells are going to be like perfect snack, and they're going to attack. And last but not least, if the same intruder invades again, memory cells help to activate the immune system to activate that immune response to make it quicker. So here we have our memory cell. It is going to now be able to know if the pathogen comes back. It's going to know exactly what it is and be able to tell the body to create or start making the antibody so we can defend the body against this pathogen that has once invaded before. So let's take a look at our overview once again. T cells can also produce memory cells with an even longer lifespan than B memory cells. Subsequent infections by the same pathogen therefore provoke a much more rapid immune response. If little or no symptoms are experienced, the organism is said to be immune. So that's where immunity can come from if you've been infected by a pathogen before. So we've talked a little bit about each of these types of cells. I'm going to ask you to pause the video, and using those last two slides, I want you to come up with a definition for each of these words. And again, we just went through each of them, what exactly they were doing. So I want you to pause the video, see what you get from the last two slides. Okay, hopefully you paused the video and got your own definitions. I'm just going to go through it really quickly just to make sure that we're all on the same page. So remember, macrophages are the ones who are going to first engulf the pathogen or intruder, and it's going to represent the antigens on its surface from the bacteria. T cells are going to activate helper T cells that are going to activate B cells to start producing plasma and memory cells. And remember, our T cells were the ones that recognize the antigen. Our B cells now are going to make the antibodies, the plasma, B cells, and it's going to remember those antibodies just in case the pathogen comes into your body again at a later time. So now, taking a look again, kind of overview of how antibodies are produced. Let's just go through and make sure that we're all now on the same page of how exactly we, as humans, in our immune response, can make antibodies to be specific to specific pathogens. So, looking at A, remember, there are many different lymphocytes. We have a lot of different ones in our body, T cells, B cells, and they are going to be reproducing if there's a pathogen or intruder in the body. B, the antigen from the digested pathogen is presented to the lymphocyte. Remember how our antigen is on the cell surface or the surface of the pathogen. C, the lymphocyte with a surface epitope complementary to the antigen is selected. The lymphocyte clones to produce many plasma cells, so this occurs in the lymph nodes. And remember, those plasma cells are making the antibodies. Then the plasma cells are going to start cloning, they're going to start reproducing. The gene for the antibody is expressed and secreted into the plasma and tissue fluid. And lastly, the antibody circulated in body fluids destroys the infectious antigen. So if you look at the picture down here, it correlates with each of the steps. So we have many different lymphocytes, okay? We are going to have that antigen be presented to a lymphocyte. The lymphocyte is going to notice that antigen. It's going to start cloning, and those plasma cells are going to start creating antibodies to be released. Those antibodies are going to bind to the antigen of the bacteria, and once it does that, remember in that previous slide, the eater cells are going to come along and eat that bacteria because they have now been recognized by the body, and the antibody helps us out with that because it's binded to the antigen of the bacteria. And right over here, antibodies, how exactly are they helping us? How do antibodies exactly kill these pathogens? Again, kind of an overview right here. So again, antibodies destroy antigens in 3 different ways. First one, antigens that are toxins can be inactivated by a reaction with the antibodies. Number 2, antibodies can attach to antigens and make sure that a phagocytic cell will recognize it. That's what we were talking about before with the eater cells. And 3, antibodies can kill bacteria by destroying their cell walls and cause the bacteria to lysis. And lysis is just a fancy word for destroying the cell wall of a bacteria or of an organism to make it disintegrate. So These are the three things that antibodies can do and how antibodies help the body by destroying the pathogens. And here we have a nice antibody production video that I'm going to recommend you watch. It's a nice overview, again, visually to see what exactly these different cells are doing, because it is a lot of information all at once with all the different cells that are playing a role. So after you watch this video, take a look at this video that's attached here. And then lesson 5 we will be doing tomorrow. So I hope you enjoyed this video. It is very fascinating and interesting material and things that we're talking about here. And just remember that for homework now, I'm going to ask you to do this worksheet based off of the immune response that we just talked about with antibodies and antigens. So if you have any questions, please feel free to ask, email me, join the Google Hangout. I am happy to help in any way I can. And also, if you'd like to say hello, feel free to join the Google Hangout. Miss all of you. I hope you're doing well, and I hope to see you soon. Have a great rest of your day.